Hi students, here in this video I am going to explain plant growth regulators. What are the plant growth regulators? In the plant growth regulators which is to regulate the growth and development. These are the things which used to come by the genetically, that is intersteric means internal cellular factors, intrinsic, they are the characters, and these are the very small compounds, chemical compounds, which is to regulate the growth and development of the plants. That is the most important thing. And they are the very simple chemical compounds and produced by the plant itself, and they will regulate the entire growth and development of the plant. Here it appears, by seeing the textbook, it may be become easy to you, but it is a little bit difficult. Here can twist it with the regular hormones. More than 30 neat bits can be formed from this one. So, so in the neat orientation, it is the most important, and it can be interrelated to other topics also, that I am going to explain in that pattern. So carefully see and listen the whatever I used to teach in this video. So plant growth regulators are the simple small different molecules, chemical molecules. They are the simple molecules. Simple molecules, why? Simple molecules only can diffuse from cell to cell, or they can send through the plasmodesmata if it is simple one, or that they are the simple chemical molecules which are produced by the plant itself, and they will be the in small amounts they will produce, but they will be very useful for the plant growth and development. What are they? He has given 5 types of the regulators here. All these 5 types are the major one. Some other things also there, but it is not a major group. It is in that one. Whenever we are seeing indole compounds, indole 3-acetic acid, and its other compounds also will come in the when you are studying the oxygen. Indole 3-acetic acid will work as the growth regulator. And next is adenine derivatives. What is this adenine? Adenine is one of the nitrogen base which is to participate in the formation of the nucleotide. So adenine derivatives. This also he has given N6 perfluoryl aminopurine, and this is called as kinetin. Cytokinins also can be called as a kinetin. That will come later on in the next video. And carotenoids derivatives. Carotenoids are the carotins and xanthophylls. They will also will produce some, that is the abscisic acid. Abscisic acid will be produced by the carotenoid. And next will be the terpenes. Terpenes with the gibberellic acid will be produced from the terpenes. Gaseous, that is the PGR, ethylene. Now, how many bits will come from this? We'll see. Secondary metabolites which are the producing the following chemical PGR, plant growth hormones. And now, what are they in this one? Now, secondary means carotenoids are the secondary metabolites, and terpenes also secondary metabolites. These two will come. You can give in the matching also, or the statement. And next will be the gaseous condition, which is PGR, that is the thing. And nitrogen base will be causes for the for producing of this following phytohormone. He can ask. You see how many types he can ask. So such type of all these things, they are called as plant growth substances or plant hormones or phytohormones, and generally we used to call it PGR. So these are the things. Now we'll see. And now the thing is, these five, whatever are there, major group, they can be their function, based on their function, positive function or negative function. When the car is going, accelerator is most important for the, but the brake is also important. So positive means there are some plant growth hormones, PGRs are there, which will be very positive for the development. But at the same time, brake like means it is to whenever the some unfavorable conditions or some emergency conditions, brake is also important. That is the one more group. So two groups they divided. First group is growth promoting. Growth will be promoted. Development will be, growth and development will be promoting. And these what they will do. And next will be the inhibitors. It will stop temporarily or permanently for the growth because of the some adverse conditions or some other things. That also we'll discuss in later. Now, in this one, growth promoting, what they will do? Growth promoting required cell division. At the same time, the differentiation, grow, and the next will be the all the metabolic activities and seed formation, flower formation, fertilization, all the things it required the some type of the PGR. So those things will come under growth promoting auxins, gibberellins, cytokinins. These are the 3 out of 5 will come under the promoting. They will be growth and development, they will promote it. And growth inhibitor, growth will be stopped or it will send the resting period in the unfavorable conditions and whenever it arises. That is also important. That in that one, mainly abscisic acid. It is called a abscisic acid. That is a negative growth. Now, one more is, now 4 are over, 3 are the promoting, 1 is the growth inhibitor. One more will come in between, they will work as the positive and the negative also. Means they will promote and they will inhibit. Both the things it will done, and that phytohormone is in the gaseous form. You see, and how many types you can ask. So both the types, positive promoting and obsession, that is inhibiting, and it is a gaseous. How many I can ask you the question in any way, in any direction. So that is called as ethylene, which is the gaseous. And majority he has given in the textbook very clearly, because it is working with the promotion and it will be the growth inhibitor. Both will come, but in its functions, majority of the functions are inhibition. That is also given clearly. It is most important for me. And next, and now such type of the, because now you remember these things he never mentioned in the Auxins, Gibberellins, cytokinins, and anywhere he didn't mention all these things. So you have to read and practice it here only. Now to come to the discovery of the this is the growth, plant growth regulators. How they discovered such type of things. They are discovered accidentally when they are doing some experiment for some other purpose. Accidentally they discovered all the 5 major groups. What are they? Auxins, gibberellins, cytokinins, abscisic acid, ethylene, which is the gaseous. So first, whenever we are seeing auxin, auxin was observed by the Charles Darwin and his son Francis. They are conducting one experiment. In that experiment, they closed a potted plant completely, and they what is that plant name? Canary grass. This is also important. And Unilateral illumination. What is the unilateral illumination? Here you see they closed a part with some darkness they produce, and this is the part, and now they given light from the side, means the whole one side, lateral side. The plant started to bend like this. Now the thing is, what did they observe? How the whenever this with the light, which direction it is coming, towards that direction the plant started canary grass, coleoptiles. What is the coleoptile? In the monocot seed you studied coleoptile, and coleoptiles used to present on the surface of the plumule, and these coleoptiles are bended in that direction. This is called as phototropism. Photo, light, tropism, bends. Phototropism, geotropism means towards the positive direction of the whatever the gravity. Phototropism and geotropism both will be done by they will show, the plant will show that one. Roots will be geotropism and the shoot system will be the phototropism. Because to get the more sunlight, the plant will bend towards where the light is come. That is the illumination. Illuminated to the unilateral illumination. Only one direction they send. In that direction only plant started. How it is, why it is, what is the cause? That I will discuss in the Auxins. So auxins are cause for the phototropism and geotropism. And next will be these when they discussed, when they observed this one repeatedly, and they told that is something is cause for this bending of the coleoptile and produced in the coleoptile, means apical meristem used to produce it. That is the thing. And later on, that he the son, he was then he has given the thing is vent, FW vent, isolated that whatever the cause for the phototropism in the plant, he isolated, but in different plant, that is the oat seedlings coleoptiles. Oat seedling coleoptiles he taken and observed. And one more thing, what the scientific name is Avena sativum. Avena test, Avena, remember it. It is out of, but we cannot say, but the Avena. What name is the Avena sativum. Seedlings coleoptiles is separated the thing. And next will be the gibberellic acid. Gibberellic acid is one more growth promoting type of the growth the plant growth regulator. And this how he observed, that he has observed, this is observed by in the E. Kurosawa, Japan scientist. He is working on the rice plant. In the rice plant, in some of the seedlings will show very elongated stem and they cannot survive also later on. Such type of the disease called as Beckney disease. That is foolish seedlings. Means they used to grow and they won't have another strength. And that is caused by also, they know, the name is, that is the Gibberella frugicori. Gibberella frugicori, a fungus, is secreting something and causes for this disease, foolish seedling formation. And it is what it is foolish, they will elongate in the seedling itself, very, very highly elongated, and they will die later on. So Beckney disease we used to call. He observed how it is coming. He taken the extract of the diseased plants, he crushed it and taken the extract, and he filtered with the bacterial sterile filter, means whatever the bacteria and other things cannot pass through that one fungus also. Such type of filter he sent. That filtrate, whatever it came, he applied on the good seedlings, which are not attacked the disease. They are also shown the same. So he told this is the cause of the enzyme which is or the something chemical which is a disease. This disease is coming because of the this fungus is secreting something. That is the cause. Not the fungus is directly. They are secreting the enzyme outside. That is the cause for this disease. Later on, they come to know it is the gibberellic acid. He didn't get, he didn't separate it, and he is not told as it is gibberellic acid. He told even though from the extract which is passed through the filters, the filtrate is also cause for this one. So the whatever the disease caused fungus is secreting something which is present in the extract itself. That he told. Later on they came to know it is the gibberellic acid. And next will be the one more, that is the cytokinin. Cytokinins, how they observed, that is the Kung et al. Means Kung and his co-workers he wrote. Et al. Means all other workers. What they done? They taken the undifferentiated tissue. From where they taken? In the tobacco internodal segment. Tobacco plant, in that internodal segment, some tissue are there which is undifferentiated after tissue. That undifferentiated mass is called as callus. Remember it. Tumor he given, no, that is callus there in the development. Here also remember it, that is the callus. So he given the auxin and nutrients what are the required for the formation of the root, stem, leaves and all the things. It means tissue culture he try to done with that one. What is that one? The callus of the tobacco which is present in the internodal segment. That is the thing. And they are not different, they are forming the callus only. They are not differentiating as the root, stem like that. Then they thought if we add something to this one, it may be differentiate. What they added? They taken the extract of vascular tissue, yeast, coconut milk and DNA. Why they taken that? That also we'll see. Now vascular tissue, he will ask the Neet. They taken and they added the extract of xylem. Then it won't work. Xylem, out of 4 complex tissue, 4 types, 3 are the dead cells. It won't be. Mainly phloem cells. Remember it, just it is out of, but for your information I am telling. And now vascular tissue, crushed vascular tissue and applied. At the same time yeast also they taken and crushed it. Coconut milk, why the coconut milk? You can take any milk, no? Coconut milk, it is nothing endosperm. Endosperm will be formed by the triple fusion, means 3x. DNA will be more. And DNA, why it is required? Because it is purine is required. Purine is required. Purine is adenine derivative. It is to present in the DNA. So, whatever the coconut milk contains of the endosperm, which is having the 3N, means 3, that is a triploid condition. So, more it will be there. So, whenever one of the extract he added all these things, then they started differentiation. The callus is differentiated by the adding of the following by the Skoog et al. He will ask that is all these. That is the vascular tissue, yeast, coconut milk, and DNA also directly separated. DNA also extracted. They added. Then the callus of the whatever the tobacco plant has started dividing, as the differentiating, as the root, stem, leaf and all the things in the tissue culture. So they thought something is there in the Whatever he has given vascular tissue or yeast or coconut milk or DNA, they are taking something in this. That is adenine derivative we already studied. That is the thing, cytokinin. That they given the name as kinetin. And Taylor et al. He has given in the new book, but old book he has given that is Cook and Taylor. They separated this thing, that whatever the compound which is using for the differentiation of the colors, they called it kinetin. See how many bits are there in this. Now, so in that way, cytokinin they found. Now, one more thing is, that is the inhibitor. Inhibitor, already I told, that is the second one, growth inhibitor, abscisic acid. But they didn't find abscisic acid with a single attempt. Three scientists individually, when they are not combinedly work in different countries also, they done on different ways, and they found some of the chemical is inhibiting the growth, dormancy, and other things. Those they told that not only independent scientists found inhibitor B. This is the most important. Inhibitor B, abscission 2, dormin. What are they? That also you should know. For the further explanation I am giving. Inhibitor B, it used to inhibit in the unfavorable condition. Means closing of the stomata, that already studied. And when the wound is formed, at that time also it is to inhibit the growth. There means that should not be this thing. And next will be the abscission 2. What is this abscission? Whenever the leaf is falling, it will be disconnected from the main plant by the abscission formation. Or whenever any place some cutting wound is formed, it will stop the sending of the nutrients or the water to that part by the formation of the abscission. That is the thing. So they found that is the inhibitor B, abscission 2. And dormin. What is the dormin? For the dormancy, seed dormancy, or the plant will undergo the dormancy in the unfavorable conditions, mainly in the plants which are growing in the very highly low temperature conditions. In the winter they cannot grow, they will go on the sleeping condition, that is the dormancy. So whenever they are showing some dormancy, at that time dormin will be produced, and these three independent scientists at the different places, they observed this one, they published a paper. Later on they came to know, scientists mean other scientists, all these three belongs to one chemical only. Only chemical is the same, but it is working in different ways. For the which ways they are working? They are inhibition, abscission, dormin, all are the same chemical, which is called as abscisic acid. A means abscisic acid. That is the thing. Next, cousin. One more is there. That cousin, what he has done, he taken some ripened oranges, means fully ripened. Those oranges, he kept these oranges along with unripened bananas in a storage room, in the store. Then they were what they done. They had hastened, means quickly they ripen. Which one? Banana ripen for condition ripen time is the become fast. Easily they ripen. He thought some volatile thing is coming out from the orange fruits, ripen fruits, and it become the cause for the early ripening of banana. Later on, they came to know that is the ethylene gas, which is the plant growth regulator. In this way, so many names, so many examples came, and they will be interlinked with other hormones also and previous chapters also. So you must be having the proper idea about this thing to get 30 bits can be formed from this one easily. And it is more names are there, so it is important one thing, and interrelated to the other independent whenever we are reading other plant growth regulators. There he has not mentioned this information, so there by mixing that one he will ask. In that way it is most important thing. So carefully listen this video and see the video and note the important points from this one. Then if he gives any bit from this one, you can give the answer. If you like this video, please give the like mark. And such type of videos up to now 41 videos I released in this one. And again I am going to release so many, nearly 80-90 videos will come. So if you want to see all the videos whenever I release in the YouTube, please subscribe the channel. You will get very good information. And if you see and if you clearly understand my videos, it is not necessary to read all the books, because it is a concept orientation. If you know the concept, you will come to know everything. That is the thing. So bye.